5 minutes, I will give you some perspectives on what I think will be the future of research for human nutrition globally, so also in this perfectly new building. But before doing so, I would like to share with you a little bit about the history of nutrition research, because it's a very young scientific discipline. It only started in the 19th century. And of course, a major achievement was, as you all know, I think it's not completely on the list, but can you make it a little smaller, because it's not, let me see. Yeah, the slides are not projected according to the screen. But I will tell you, of course you know this person, Professor Christian Eijkman. He was the discoverer of the well-known first vitamin. The first vitamin was discovered in 1910, so a long time ago. It was vitamin B1, thiamine, and all the experiments together with Professor Grijns, who is from the Wageningen University at those times. They did all the nice chicken experiments and discovered the crucial vitamin, the first one of the whole vitamin revolution in 1910. And he received the Nobel Prize for this, as you all know, in 1929. By the way, he was born, Professor Eijkman, in Nijkerk, in the Netherlands, and Nijkerk is about 30 kilometers from Wageningen University, which is interesting to know maybe. So there was the vitamin revolution, and then really the research on nutrition started. And then in the 50s we had, you know, the start of the knowledge about nutrition and cardiovascular disease, where this person, Ansel Keys, was really very fundamental in, let's say, discovering the role of fat and saturated fat in heart disease. And you see the Times magazine, the cover in 1961, where it was yourself mentioned that he did all these discoveries. But you can see in the Time cover in 2014 that the whole issue of fat and fatty acids is still keeping us busy in science. But also from the 60s onwards, we saw a dramatic increase, especially in the US, but also in other countries, in the obesity epidemic. It doubled from 1960s, let's say from even the 1980s, so in the last 30 years, it doubled in the Netherlands, and now in the US, as you know, 1 in 3 Americans is obese, a huge problem. But let's say also in the same time period, the hunger rates increased worldwide. And what we also have seen in the last 15 to 20 years, that we see the double burden of malnutrition, where under and overnutrition coincide, so are present in the same individual, especially also in Asia and Africa, where you have a real double burden of disease due to malnutrition, and I will come back to that. The knowledge on how diet plays a role in health was really very strongly stimulated by the epidemiology, nutritional epidemiology, but also by all kind of very interesting dietary intervention studies, controlled intervention studies. We learned a lot about the role of nutrients, of foods, of complete diets, and we know right now that saturated and trans fats are bad for you. Too much salt is bad for you. Added sugar is a real problem everywhere in the world. And of course you need to eat sufficient fiber in order to achieve a balanced diet with good life expectancy. What also happened, and then I'm talking about, let's say, the beginning of this century, where of course the human genome was mapped, and also the mapping of the epigenome, which brought us a complete new insight on what's genetic susceptibility and also gene expression would mean in relation to diet. And then the new buzzword came up, personalized nutrition. So what does a good diet mean for me personally? So the research is massively, let's say, working on personalized nutrition. I will come back to that later in the talk, but it really is a very interesting topic, but very difficult to study. So that will really require some decades before we have made major achievements. Another thing what happened is, and this is in fact also crucial for nutrition research, but for medical research, any research in general, that we nowadays can collect data as much as we like in all different ways, with smartphones, with all kind of devices, wearables, with MRI scanners. You can measure anything on an individual level, but and of course it's very important to do so, but the difficulty here, and we need good bioinformatics, we need good statistical methods to work on the use of big data in nutrition science as well. I will now go to address, as I said, perspectives for the future for nutrition, and I have selected through 3 stages of life: early life nutrition, midlife nutrition, and late life nutrition. But because of the time, I have to skip late life nutrition because I will not manage in the next 50 minutes. But I would like to give you a flavor on early life nutrition and midlife nutrition. On the one side, I will give a few examples of the past, and I will give a perspective for the future. Early life nutrition, that's the first topic. If we look at this map, we can see in yellow the 34 countries in the world where 90% of malnutrition occurs, which mainly relates to calorie deficit, not too much protein, and of course the hidden hunger, the big five micronutrients being iron, vitamin A, folic acid, zinc, and iodine. So in these countries, especially concentrated in Africa, in Asia, we see this high burden of malnutrition. And if you imagine that nowadays that malnutrition is responsible for half of child deaths under the age of 5 worldwide. And this is a figure we simply cannot accept in 2016. And we need to find different ways to tackle this huge problem. In Wageningen, we are collaborating, as is seen here by different dots, red dots, with several centers in this region to tackle and to study, and also to work on the capacity building in those institutes on malnutrition in the world. If we talk about malnutrition, we have, in fact, especially for micronutrient malnutrition, we have 4 solutions. We can fortify, we can biofortify, we can supplement, and the 4th one is the dietary diversification. I cannot address them all, but I would like to give one very interesting example studied by Dr. Rina Agustina. She's taking always pictures, she likes that, Rina. Rina Agustina, she did her PhD with me in Wageningen on the impact of probiotics and calcium on diarrhea and respiratory tract infections in Indonesian children. It was a huge undertaking with a lot of logistics and organization. It was a 6-month double-blind, placebo-controlled trial in almost 500 healthy children. 10 minutes left. Oh, I see it here. And healthy children aged 1 to 6 years old. I cannot go into you too much in the details, but I can tell you that this was a huge undertaking. And Rina, she published very nice papers, several papers, but one on the incidence of diarrhea. So these probiotics were a specific Lactobacillus reuteri strain, and it can be seen from the proportion of children without diarrhea indicated by the 4 lines. So there was the ones who got the Lactobacillus reuteri, it's the red line, they had less diarrhea, but it was not statistically significant, but there was at least a very clear trend, especially in children with a lower nutritional status. And we also saw that regular calcium milk, because they were delivered milk, and in this milk was either a low calcium or high calcium, as well as reuteri or no extra Lactobacillus reuteri, so it was a 2x2 factorial design. And it could show that regular calcium milk alone or with Lactobacillus casei, that was the other lactobacillus, did not reduce diarrhea, which was in fact a disappointing finding, but at least those children with a low nutritional status benefited, and this is low nutritional status is quite common in children in Indonesia. What was very important is the launch of the 1000 days. This initiative was launched about 6 years ago in Washington, and it indicated, it really was a movement to make clear that the first 1000 days of life are crucial for the child's future perspectives. And it has a lifelong impact. And this initiative is really stimulating public-private partnerships to look specifically at the first 1000 days of life. And these 1000 days of life, I can illustrate how important if you look at iron. Iron is very crucial for your brain, and especially in the last trimester of pregnancy until the age of 2 of the baby. It's really very crucial that your iron status is sufficient, is optimal, because otherwise it might not only result in anemia at birth and later on, but it is also shown in animal experiments that it also might have a strong impact on your mental and cognitive development of children. And I can show this by a paper which was a Chinese study, another PhD from myself in Wageningen, Xuying Chang from China, and she looked into the iron deficiency and also in the first 1000 days of life. And what she could show is that this was again not a 2x2 factorial design, but a normal supplementation study where 7,000 pregnant women received during pregnancy extra iron with folic acid or folic acid alone. And you can see that the infant mortality with extra iron was significantly lower after birth. And also there was less anemia, there was not a lower birth weight, there were all kind of other benefits of this extra iron during pregnancy. And what she also could show that later on, that these children after 2 years, she could show that these children had very good cognitive development unless they had their extra iron during pregnancy. Important point is that the first 1,000 days are an important critical window to combat malnutrition. But maybe even more important is the age of the adolescent girls and adolescent girls' nutrition. Because adolescent girls, you see quite a high prevalence of pregnancy. There's a lot of stunting also in this age group, and there is full strong growth in these adolescents. And in fact, what they are eating is not only influencing their health, but also the health of their children and even of their grandchildren. And this is related to epigenetic. And epigenetics is in fact really understudied. It should have much more attention in nutritional studies, the role of epigenetics on your health. And epigenetics refers to the situation that you have effects on the gene expression, and the effects on the gene expression may be because of the impact of methylation of the DNA. This is a major impact that can occur, where in fact the genes are not sufficiently expressed due to methylation, and this methylation is influenced, of course, by the diet that a pregnant woman are using. And there has been a very nice study published in the Nature last year, where it could be shown that in a situation where mothers were pregnant in a hungry season, of course with not a lot, with a bad diet and not too much methyl groups, compared to the harvest period where there's sufficient food, that you could see that the children had really a bad health, and because of epigenetic changes that occurred because of the less good diet during this hungry season. And I think that this type of studies we need more in order to learn more about how the epigenome influences, in interaction with the diet, influences the health of young people. And we need to study this specifically, I think, in adolescent girls, in adolescent nutrition, because that's a very crucial time window to combat micronutrient deficiencies as well. Very important is this slide to show a worldwide map what the prevalence is of stunting, of reduced growth in children under 5 years. And you can see the prevalence rates indicated by the colors. And important, what I would like to mention as well, of course, nutrition-specific solutions has through the 4 ways I mentioned, fortification, biofortification, supplementation and dietary diversification is important, but nutrition-sensitive approaches are crucial as well. And these very crucial nutrition-sensitive approaches can be seen in, and it was a very nice paper last year in the Lancet. I really advise you to read this, that, for example, with respect to good sanitation, to good hygienic measures, agriculture, empowerment of women. These are nutrition-sensitive approaches which, in combination with nutrition-specific approaches, are probably much more effective than only focusing on nutrition-specific. So I would advise you to also take these nutrition-sensitive topics into account. I saw 0 minutes,
2 minutes.
2 minutes left. I would like to cover with you the midlife nutrition as well, but I'm afraid that I don't have sufficient time for it, because at least I need 10 minutes. But let's try to find one or few of the major slides, at least to give you a flavor. Tomorrow I will give a guest lecture at the institute here, so I will cover all three, so early life, midlife, late life. But now I can, obesity is a very big problem worldwide, and the environment, of course, diet is a factor, but it was again a very nice paper in the Lancet about obesity, where they were stated that especially the environment is crucial, because there are, advertisements, there is the provision of not very healthy foods, and people are indoctrinated, in fact, to eat and to consume as much as possible, which doesn't help so much for weight management. Important is to tell that already 5% weight loss is sufficient. These weight loss programs, either working on 5 minutes I have. I saw 5 minutes. So either looking at more proteins in your, when you take a weight reduction program, people are saying you should eat more proteins, that's good, to lose weight, because they are saturated, they give a satiety feeling. Others say you have to reduce your carbohydrates a lot, or fat. It doesn't in fact matter what you choose, it's in fact the calories that count, and the calories determine how much weight you lose. And if you look at the meta-analysis of all the trials that have been done on weight loss, they are very disappointing. I mean, the immediate benefit is okay, but in 3-4 years, all the benefits are gone, almost always. But I have to mention that already 5% weight loss is important, because that is already giving you health benefits. I have no time, but I would like to use, let's say, the fact that we nowadays can measure at an individual level, at all different stages in the diet, in the epigenome, in the microbiome, in the metabolome, the proteome, in the transcriptome, etc., we can measure a lot. This is in fact big data, and the question is how to deal with all these data. It's important because we can learn much more on an individual level. What are the risk factors? What is maybe helpful? How are the mechanisms? And thereby we also might find more specific clues for solutions on a personal level. But as I mentioned in the beginning, it's extremely difficult to integrate all this information, but we have to. That's the future of nutritional sciences. I wanted to spend, but I will skip this, I wanted to spend a few slides on a personal, let's say I was a guinea pig myself. I did a self-experiment of 2 months, eating during 1 month, and you see here what I was eating during 1 week, an unhealthy week and a healthy week, but I followed a healthy pattern 1 month and an unhealthy pattern also 1 month. Of course, doing all the bad things in eating and not exercising a lot. We measured a lot of stuff, from body scans to of course, blood measurements, wearables, so a little bit a multi-level approach. And really, it's indicated here. I was completely disappointed. You can see it in my face. What unhealthy changes occurred in me already after 1 month. I'm quite eating healthy, I would say, as a professor of nutrition. I started quite healthy, but after 1 month, I saw worse blood lipids, increased insulin, higher liver enzymes, etc., etc. Of course, this is not, let's say, science, but because it's only N is one person, but I really was very much convinced how important prevention can be and how important also the multi-level of data that you can collect and you integrate in order to learn about the health of an individual. That's very important. And what is important is nowadays, if you see this person, he has a BMI of 36.2. And all the fat, it would mean, in fact, that this person would be 40 kilograms heavier than I am. The problem is that the fat, all the fat that this person is accumulating is located in the abdomen, and especially within and between very important organs like the liver, etc., etc. And what can happen is that in this situation, that these cells are, the adipose tissue cells, they are increasing, and also the cells in the abdomen, the fat cells are increasing. They can expand almost 20 times their size. And what is happening is that they are, let's say, releasing specific factors that can connect, that can interact with the immune cells. And these infiltrated immune cells, they are, let's say, then located into the adipose, and they can give chronic inflammation, leading to subsequent insulin resistance, and later on diabetes and other bad consequences like heart disease and even some forms of cancer. The time is up here. I will not address this one. It is clear that this is very complicated, but in order to learn the mechanisms, how the adipose interacts with the immune cell, I mean, that's a lot of molecular biology, which if we know more about this, we can use it for pharmaceutical purposes and of course also for nutritional solutions. My last slide, I will not address this very interesting study. It was very interesting. We measured a lot. As you can see, we even measured also in the brain satiety after weight loss. We used, in fact, a very nice device to sample gastrointestinal content, where we could study microbiota. Sorry for that, I cannot. We used a lot of, of sophisticated ways of studying the diet, and this is, in fact, where it's all about to integrate all this multi-level data and to learn what exactly is happening within an individual. Because in this weight loss trial, there were some people losing on the same diet and the same regimen. We were using 8 kilos and the others were only 2 kilos. So what was happening? And if you have all these data and you integrate the data, you can understand better why the one is successful and the other is not successful on the same regimen. And that can be used, and this is my final slide, this can be used in our scientific approaches to go from a one-size-fits-all, which in fact now is nutritional science. We are talking about an average person. We can go to subgroups that might be based on all these big data, that might be more, let's say, successful in losing weight. And we even in the future could go to an individual level, and there we are very close to personalized nutrition. But as I mentioned in the beginning, it's a long, long road. Thank you very much, and excuse me, I did not have so much time.